Hi, I'm Caroline LePoole, and I want to talk to you today about modeling microbial impact on vitiligo. And we test this primarily in mouse models, but I will also give you some idea of what we're working on in human patients with vitiligo. So I have a lab in the Lurie Cancer Center at Northwestern, but also have co-direct the tested core for the dermatology SBDRC, and I'm the CSO of a small company called Dietary and therapeutics. That will not be covered in today's talk, however, so there should be no conflict of interest to be concerned about. The hypothesis we are testing then is whether dysbiosis can serve as a trigger for vitiligo induction. Not only that, but we're also interested in seeing if microbial compounds could serve to perhaps keep depigmentation in check. Working according to the concept that pathogenic microbes can find their way through the epithelium, either in the gut or in the skin, and then activate dendritic cells, innate lymphocytes, and finally antigen-specific T cells to trigger a response to melanocyte antigens that can lead to depigmentation. Of course, the opposite can also occur, especially if we think that this might trigger a greater regulatory T cell response to keep vitiligo in check. When it comes to human patients, we haven't gotten that far along, especially as we have a lot more samples than we have money to analyze the samples. But we've performed shotgun metagenome sequencing on 5 samples of human patients compared to controls. And we found outcomes that jive quite well with the publication we've all seen in Journal of Investigative Dermatology earlier this year, which I think is really interesting. And you'll see this come back also in our mouse studies, that there seems to be a greater prevalence of Bacteroides in controls, which could be of interest if this helps to keep vitiligo in check. But right now, the one significant difference would be a greater prevalence of Parabacteroides for these 5 samples that we looked at. These Bacteroides that we are kind of focusing on for the moment, if we look at them in more detail, we can see which species are particularly overrepresented in controls compared to patients, and that would be Bacteroides vulgaris, fragilis, or plebeus as some candidate species to look into and see if there's compounds of these bacteria that could be important for halting depigmentation. We're not only looking at the microbiome itself. Actually, we put a couple of questionnaires in front of the patients when they come in, when we also sample their skin microbiome. And we so far did not find any correlations to duration, FASI scores, BSA, or VITI-QOL, but we did see some other parameters that were somewhat modified in patients compared to controls. Specifically, we find that patients are more likely to be affected by hypercholesterolemia and anemia. This is something we've kind of reported earlier as well, especially the anemia. There's a greater odds ratio among patients. And what's interesting is that some of these factors are found to precede vitiligo. We reported this in our earlier publication. So that keeps us thinking as to whether this might contribute to disease development rather than be merely a consequence of a different appearance. We also find them to be more frequently associated with depression and anxiety, and to be more frequently treated for these conditions. What stood out to me otherwise was that there seems to be a tendency to bathe less frequently and shorter than controls. Patients also apply sunscreen and makeup more frequently, but that shouldn't be much of a surprise. In general, however, it makes me think that patients are more careful about their skin than controls. And at the same time, we find that vitiligo subjects are more likely to be affected by other autoimmune conditions, which are listed here. Now, Hashimoto's should not be much of a surprise to you. Back to the microbiome, if we think of composition of the microbiome in the gut, we would tend to think about the potential consequences of food intake and then especially nutrients that could affect microbial growth. And so we wanted to look at the Healthy Eating Index. That's a very lengthy questionnaire developed by the NIH, the NHANES test. And we can derive from this Healthy Eating Index and found for the first 10 patients and controls that there was really no overall difference in how healthy patients eat as compared to controls. But we can look at some of these composite scores, and these might lead us to some conclusions in the end once we have analyzed a greater patient number. So far, it looked like there might be a greater sodium intake by patients than by controls. Back to mice. The nice part is that mice can be included in studies where we can look at cause and effect more easily. So what we did here is to do a antibiotic study. Antibiotics are, of course, known to affect the microbiome, and for that reason, we were interested in whether that would have consequences for depigmentation that we observed in the mice. We took the oldest known spontaneous model of vitiligo, which is the FH mouse that was obtained from the Engelhard lab. The mice developed depigmentation in response to a transgene encoding a T-cell receptor against tyrosinase that recognizes melanocytes based on a tyrosinase peptide presented in the context of HLA-A2, so they are also transgenic for that MHC molecule. Pregnant females were then supplied with antibiotics in their drinking water, and after birth, the pups were also maintained on the same antibiotics. So essentially, the pups were never really able to set up a healthy microbiome in the gut or skin. And so we can scan these offspring over time by flatbed scanning, and what we then found was that there was a much greater development of depigmentation in response to ampicillin. And if we look at the ampicillin-treated mice, we see that their gut is inhabited by much greater proportion of Pseudomonas in the gut, and rather consistently with perhaps one outlier here, and that the neomycin-treated mice also had a much different microbial composition as compared to control mice, with much less diversity. And in fact, Bacteroides are winning the game here. And that's interesting because even though this difference is not yet significant, there is a trend towards a reduced vitiligo development in neomycin-treated mice. In fact, there was really no significant depigmentation to speak of in that treatment group. Depigmentation was associated with a greater prevalence of IL-1A and CXCL13 transcripts in the colon, as measured in homogenates of the gut. And in the serum, we find a greater prevalence of IL-27 and IP-10, which is CXCL10, in the serum of our neomycin-treated mice. And that's interesting because, of course, we think of CXCL10 as something that can attract T cells to the skin. But what we're thinking here is that because this is actually in the serum, that perhaps it actually leads to less infiltration of the skin. At the same time, if we look at the skin, we find less T cells in the skin overall in ampicillin-treated mice, and that might surprise you, but think about this. The mice were already 30 weeks old, and most of the depigmentation is already behind us. And in fact, we'll come back to that in a moment, but the T cells are quite exhausted in the ampicillin-treated mice. Whereas if we look at the neomycin-treated animals, we find that the prevalence of T cells is quite similar to that in control mice, but the composition is quite different. So we see a much greater portion of Tregs in the neomycin-treated mice. So again, that would lead us to believe that if we have more Tregs in the skin after neomycin treatment, that might lead us to slower depigmentation, if at all. And, that makes it really interesting and something to follow up on. As mentioned, in response to ampicillin treatment, after 30 weeks, the T cells are completely exhausted and do not respond by generating interferon gamma or IL-17 in response to peptide, which is not the case in response to neomycin, by the way. And, that is something to be studied in more detail, because obviously we would expect that that would lead to greater and not less depigmentation. Then that in overall does lead us to want to study what these antibiotics would do upon topical application, because that would lead to a very practical application that could be of interest to patients. So we shaved the mice on their backs and applied bacitracin to one side and Vaseline on the other, or otherwise Neosporin on one side and Vaseline on the other. Our control mice received Vaseline alone. Again, we scanned them on the flatbed scanner and waited over time for depigmentation to occur. In this case, we do see a significantly delayed depigmentation in the case of mice that were treated with Neosporin alone. Keep in mind that this is a different model. These are female mice. They respond to T cells that recognize GP100, but the principle is the same. And interestingly, if we look at both sides of the mice that were treated with either one of these antibiotics or neither, we see that there's great concordance between depigmentation that occurs on both sides of the mice, which would tell us that there might be some abscopal effects going on if mice are treated with Neosporin, that their other end is also protected from vitiligo development. The topical application of antibiotics did not impact the composition of the gut microbiome. Their composition was similar when we looked at each one of these genera. And so obviously, we think that there's more likely a change in the skin microbiome in these cases where we applied the antibiotics directly to the skin. And the skin microbiome has not been tested yet, and something we want to do shortly. But in summary, what we're looking at is gut colonization by Bacteroides associates with better disease outcomes. Patients and controls mainly maintain a similar diet composition, or at least the health of their diet is no different. But the composition of individual compounds may be different. That's something we have to figure out. Topical interventions may serve to combat disease, and patients tend to be more careful about their skin. Now, I want to acknowledge especially the contribution of the clinical part of our department, led by Dr. Rupal Kundu and also by Dr. Stephanie Rangel, who have been able to provide us with samples and also with clinical data for the patients that we're interested in. And my own lab has been actively involved in these studies. Right now, it's mostly on Rohan's shoulders, and we collaborate with Loyola, where I started these studies together with Catherine Knight, and Suhail has since moved on to his own lab in Missouri. And at Rush University, Stephen Green has been very much involved because he designs all the microbiome analyses. I'm now collaborating with the University of Michigan, Johan Gudjonsson, for follow-up studies, and our mice were obtained from Vic Engelhardt. Thank you very much, and I hope you have some great questions and that you enjoy the rest of this meeting. Thanks. Bye.